Hello and welcome. Today we're going to have a discussion about the Prometheus Panta and how it provides multi-parameter stability data that helps you assess the impact of conjugation on thermal and colloidal stability of your ADCs. I'm Dr. Rebecca Hood, and as part of my job as Prometheus product manager, I speak to a lot of customers working in the biologics field. Over the years, I've learned a lot about your most common concerns in the lab. So today I'll be sharing some data from the Prometheus Panta to show you how you can perform high-quality stability measurements in parallel to understand how conjugation impacts your ADCs, and I'll be discussing some considerations that can help to optimize ADC stability. Now, as most of us are probably aware, an ADC is a monoclonal antibody connected to a small molecule drug by a linker. These molecules have a lot of therapeutic potential, but the addition of the small molecule drug can negatively impact the stability of the monoclonal antibody in a number of ways. First, the proximity of the drug to the monoclonal antibody will impact its chemical environment and solubility. While many monoclonal antibodies are fairly easy to solubilize, the same is not always true of small molecule drugs. Next, the conjugation process itself may require harsh chemicals or buffer limitations that destabilize the antibody. The antibody must also incubate in that potentially destabilizing environment for some time to ensure that the conjugation reaction takes place. Now, once the conjugation is complete, the whole sample must be cleaned up to remove any unconjugated drug, aggregated protein product, and to exchange into a more stabilizing buffer. And of course, the final buffer required for optimal stability of the ADC may be different compared to the unconjugated monoclonal antibody. Now, if you find that conjugation destabilizes the structure of the molecule, it may be worth investigating additives to further stabilize the final ADC. Now, developing, formulating, and characterizing monoclonal antibodies is a challenge in and of itself. But with these additional risks to the stability of the monoclonal antibody due to conjugation, ADCs have even more potential for destabilization and failure. Now, like any other drug, ADCs need to be completely and fully characterized for safety, efficacy, and stability before they can go to clinical trials, let alone be approved as therapies. So today I'll be focused on stability attributes for early phase screening of ADCs and how to generate the data you need to make decisions about which conjugation approaches or candidates are the most viable for your pipelines. So by the end of this talk, I hope you'll walk away with some insights into how the conjugation of a drug to an antibody impacts its stability and how to characterize that using multi-parameter stability data. Now, one way to do this is by using the Prometheus Panta, which is a great tool for learning a lot about your ADCs in just a short time. Today, I'll walk through the considerations that affect the stability of an ADC and show some real data from the Prometheus Panta to illustrate what to look for when mitigating risk during ADC development, and to emphasize why having the best possible data quality ultimately saves you time and effort during development. So before we dive into the ADC information, I'd first like to introduce the Prometheus Panta. So the Prometheus Panta has 4 integrated technologies that work in parallel to give you deep insight into the thermal, chemical, and colloidal stability of your candidates. Let's talk a little more about what these technologies are and how they provide stability information. NanoDSF uses intrinsic fluorescence from your proteins to monitor their thermal or conformational stability. Comparing the thermal stability profile of an ADC to that of the unconjugated antibody tells you whether the conjugation reaction or the drug linkage itself has significantly destabilized the antibody structure. DLS tells you about the size of particles in solution. It's helpful for identifying if your cleanup process wasn't complete, or if there is unconjugated drug remaining, or if there are aggregated populations in the prep. It's also used to predict propensity for aggregation or viscosity at higher clinically relevant sample concentrations. A back reflection measures turbidity of a sample. It's great for identifying large amorphous aggregates contaminating your sample, and it's an early stage indicator of subvisible particle presence. And finally, SLS measured along a thermal denaturation ramp gives you very precise insight into when particles start growing in size, which could be due to unfolding or small aggregate formation. Now, with these 4 technologies, Panta provides highly precise measurements without extrinsic labels. It accommodates a wide range of sample concentrations, going as low as 10 micrograms per mil, while using just 10 microliters of sample to give you full stability picture of your candidates. So the Prometheus Panta gives you high-resolution, high-quality data on your candidate's stability. You can use it to triage the worst candidates or formulation conditions from a screen by eliminating those with poor stability attributes, or use it to create an in-depth, highly detailed characterization of your candidate and formulation for developability profiling. It works on all types of proteins, so it's a great tool even if you're not working with ADCs or monoclonal antibodies. And it's so easy to use. When we do our Panta installations, we can get the instrument running, train the lab, and have people generating their own data and learning new things about their constructs all in the same day. So now that we've discussed the Panta, let's get into ADC characterization. As I mentioned in the beginning, there are added destabilizing risks when it comes to engineering ADCs, which is why it's important to have detailed information on their conformational and colloidal stability both before and after the conjugation reaction. Now, the engineering of the Prometheus Panta lets us follow the complex unfolding behavior of a biologic, such as a monoclonal antibody, across a thermal denaturation gradient. By collecting the thermal and colloidal stability in parallel from the same samples along the entire thermal ramp, it's possible to follow the different stages of unfolding in minute detail, giving you the most possible information about your candidates. So here's an example of how multi-parameter characterization works in ADCs. Trastuzumab is a monoclonal antibody for HER2-positive breast cancer. And trastuzumab emtansine is an ADC derived from it, which is conjugated to DM1, a cytotoxic molecule. And for this talk, I'll be referring to them by their brand names so they're a little easier to say. So the monoclonal antibody is Herceptin, and the ADC is Kadcyla. Now, both of these have already reached the clinic, so they're both the optimized final formulation. The data we'll look at in a little bit is going to look optimal in terms of stability, but I'll talk through some things to watch out for when you're working with less than ideal candidates. And we'll look specifically about how the data enables us to see how conjugation impacts stability. The Prometheus Panta provides data that is high resolution and very reproducible. This allows you to detect even the subtlest differences between candidates when you're selecting ADCs from a library or have tried different types of conjugation strategies. So now let's look at some data. In the examples I'll be showing, Herceptin will be in the dark blue with Kadcyla, the ADC, in lighter blue. So the top view here shows the nano-DSF profiles from triplicate runs of each sample. Now, it may be hard to tell, but I have the individual data points from all 3 runs overlaid. The top run is in color, while the other runs are grayed out. So here we're using thermal unfolding to measure the TM, or melting temperature, which is the point at which 50% of a protein or domain is folded and 50% is unfolded. Now, the bottom plot is the first derivative view, which makes it easier to spot the inflection points. And as you can see, our overlays are still very similar to each other. There are 2 transitions here, TM1, which corresponds to the CH2 domain, and TM2, which is both the Fab and CH3 domain. Now, these domains often unfold at very similar temperatures that we can't really resolve with nano-DSF. But let's dive into how the Panta's data resolution helps you learn about the impact that the conjugation process has on your antibody stability. So with the resolution you get from the Prometheus Panta, it's easy to create a profile of different constructs and compare them to see just how conjugation impacts the overall stability of the molecule. This is the same data we just saw. The dimensions of the plots are just a little different. So since the drug conjugates to the Fc domain, the bigger impact is on TM1, which has a bigger change between Herceptin and Kadcyla compared to TM2. Now, both these temperature decreases are statistically significant, but modest, which shows how optimizing your drug before going to clinic is valuable to have minimal impact on thermal stability. Now, in the first derivative view, it's easy to spot how conjugation impacts overall stability of the first melting temp. You can easily see how the peak is broadened. So not only is there a decrease in the TM to indicate destabilization, there's also a more gradual unfolding of the CH2 domain, as indicated by the reduced slope at the inflection point. So the level of resolution from the Prometheus Panta lets you see not only how much the TMs shift and whether the shift is large or modest, but also gives insight into the rate of unfolding. So we see that the ADC unfolds more gradually and the onset is earlier, which is an additional indicator of reduced stability. But thermal stability isn't the only thing that matters. The Prometheus Panta provides you with a lot of other information that you can collect on your ADCs that will give you even deeper insight into their stability. So there are lots of parameters directly measurable by Panta and several more that can be derived from the data. Now, I'm going to focus on just a few, which are highlighted here, to show how easy it is to build in-depth stability profiles of your ADCs. Isothermal DLS measurements are really critical for determining whether there are contaminants, unconjugated drug, or aggregates in your sample. These measurements are a reflection of how your biologics are behaving on the bench. So shown here is a measurement that was performed at 25 degrees C before the thermal denaturation gradient we previously saw. So this is data derived from size analysis with DLS, which informs on the size of molecules you've got in solution, as well as the distribution of particle sizes. So you can see here, the overlay and distribution between the naked monoclonal antibody and the ADC are very similar. The ADC in light blue has a very slight change in hydrodynamic radius of 0.1 nanometers compared to the naked monoclonal antibody. This isn't statistically significant since DLS can't always resolve the sub-nanometer differences associated with the change in hydrodynamic radius that results from drug conjugation, but it does show that there is some overall change to the structure of the molecule. And what's also important to note here is that the peak does not broaden. And so peak broadening for intensity distribution profiles implies destabilization, as there are multiple populations present that are close but not identical in size. So if your peaks are broadening, there are slightly unfolded but not yet aggregated or unfolded particles in your mixture. You can see that there's a very small peak at high hydrodynamic radius. Larger size particles are overrepresented in these intensity distribution plots, but this is noteworthy because there's no corresponding peak for the naked monoclonal antibody. And last, I'd like to remind you that if your conjugation process did not completely use all of the small molecule, or if you don't have a sufficient cleanup process after conjugation, DLS can resolve particle sizes as small as ½ nanometer in size, which means they will likely pop up as an additional peak on the graph. Now, again, these are already optimized formulations, so we wouldn't expect to see that peak, but it's something to be aware of as you're developing your ADCs. So overall, there's not a huge difference in the DLS profiles of ADC versus Herceptin, and this is a good thing. As with the thermal stability profiling, it's expected. Both of these biologics have been optimized for clinical use. However, they do start to hint at what to look for in the discovery and development phase. So next we have an example of chemical stability using a chaotrope to denature the protein rather than a thermal gradient. Both the antibody and ADC denaturation is fit with a 3-state model, as similar to thermal denaturation, the different domains are impacted separately by chemical denaturation. Now with chemical denaturation, you can determine the delta G of unfolding. So basically, the more denaturant that's required to unfold the protein, the more stable the protein is. So if we compare these values, the first delta G for the ADC is decreased compared to Herceptin alone, and this goes from about 26 to 21 kilojoules per mole. For delta G2, there's a more dramatic decrease from about 81 to 71 kilojoules per mole. This is all to say that if you choose to investigate the chemical stability of your ADCs during development, you want to look for minimal impact to the delta G. So all the data I've shown you so far comes directly from the Panta analysis software. But let's take a closer look at how easy it is to identify trends or interesting phenomena with easy visualization of your data. So here's one view of the information we collect in parallel. The top 2 plots show measures of conformational stability, and the bottom 2 are measures of colloidal stability. By measuring conformational and colloidal stability in parallel, you get insight into how domain unfolding specifically impacts aggregation. So I talked before about how we can resolve the unfolding of different domains. If we look specifically at the TM2 peak here, we can also see that aggregate formation coincides only with the unfolding of the Fab and CH3 domains. So to get a clearer picture of colloidal stability, let's look more closely at the aggregation data. So now we're only looking at the colloidal stability information gained from back reflection and light scattering, and this is the bottom half of the figure that we just saw. Back reflection, which is the turbidity data shown at the top, shows a modest decrease in colloidal stability. Though the shift is small, slightly only a degree, it's statistically significant. Conjugation has a small but measurable impact on colloidal stability. Using light scattering information in the bottom plot, which is great for picking up small size changes associated with small aggregates or increase in unfolded population, we can see that the ADC starts unfolding much earlier than the monoclonal antibody. Picking up on subtle differences like this is really critical when you have a huge library to select from during the early discovery phase. So here we're looking at the same ADC we were previously looking at, Kadcyla. Now we're comparing it in PBS, a buffer commonly used during the triage process, to an optimized buffer ultimately used to store and ship the ADC. This is important because during first pass screening, you're likely only looking at the construct sequence, impact of conjugation method, or screening for different drugs conjugated to the antibody. Once you choose the most likely candidate, another round of work is required to find the optimal buffer conditions for storage and shipment. So at the top, we've got our nano-DSF data again. You can see the ADC in PBS is in gray and the ADC in the optimized buffer is in blue. Now we see a very slight TM change with the optimized buffer, but this isn't significant for either TM1 or TM2. But, and I think it's obvious when we look at the colloidal stability information, we see a huge change. In the optimized buffer, there's no longer any formation of large aggregates, which we see with the turbidity information from back reflection measurements. Light scattering reveals only a slight change in the overall cumulant radius for the optimized buffer throughout the duration of the experiment. Now, scattering does pick up on some small aggregates, also near the second TM, but they're not forming the large tangles like we're seeing with PBS. Now, from this example, we see that it really takes the power of conformational stability with colloidal stability information from back reflection and light scattering to give a full picture of what's happening with your ADCs, whether you're comparing them to a naked monoclonal antibody or trying to optimize their formulation. Now, to wrap up, I want to offer some practical solutions for reducing the destabilization risks of your ADCs. So when thinking about the practical considerations of building an ADC, it's critical to consider the stability of the antibody throughout the duration of the conjugation process. If you're starting from scratch, be sure you fully understand how the unconjugated monoclonal antibody behaves, both in the formulation and conjugation buffers. Remember that both the drug and linker will change the chemical environment of your monoclonal antibody, so it's important to monitor how these are impacting stability. And if you monitor the stability of the monoclonal antibody in the cryo conjugation buffer and find that it's highly destabilizing, you can play around with the conjugation strategy, the incubation time, and even the linker length to minimize the impact to the immediate chemical environment of the antibody. Now, of course, it's not always possible to have complete flexibility when building a new ADC, but go in armed with as much knowledge as possible about the monoclonal antibody stability before conjugation to minimize the destabilization of it during conjugation. And the Prometheus Panta provides a tool to do just that by giving you an instrument that provides a lot of information about your ADCs from just a single experiment. High-quality, reproducible data gives you confidence when ranking candidates in formulation, and the Panta's ease of use, scalability, and flexibility means it can easily meet your research needs. So as you've seen, the Prometheus Panta provides quality multi-parameter information about ADCs to help you ensure that you build a stable molecule with good developability profile. And with that, I'd like to thank everyone for joining today. It's been really great sharing with you how the Prometheus Panta can provide you with all the stability characterization data you need to make confident decisions about your ADC pipeline. And I'd especially like to thank Dr. Andreas Langer for providing all the data that I showed today.